The two vital structures that store the genetic information are DNA and the RNA. To maintain the continuity of life, it is crucial that they follow a structural framework that is replicated from one generation to another. The basic building blocks of these nucleic acids are nucleotides, which are composed of a phosphate group, a 5-carbon sugar, and a nitrogenous base. For DNA nucleotides, they contain deoxyribose sugar with 4 possible bases: cytosine, guanine, adenine, and thymine. Meanwhile, RNA nucleotides contain ribose sugar with 4 possible bases: cytosine, guanine, adenine, and uracil. One of the main components of both nucleic acids is the sugar-phosphate backbone. Nucleotides form covalent bonds between the 5' end of a phosphate group to the 3' end of a 5-carbon sugar, extending itself in a fixed orientation. Ultimately, this results in one strand of RNA and DNA. For double-stranded DNAs and RNAs, there are 2 parallel strands that extend in opposite directions. That means that one strand is synthesized from 5' to 3', while the other is synthesized from 3' to 5'. This anti-parallel nature of DNA creates the iconic double helix shape. Keep in mind that while RNAs are generally single-stranded and DNAs are generally double-stranded, there are still some exceptions. However, the structure simply would not be complete without the second main component, complementary base pairing. Found on 2 strands of a DNA or a strand of DNA and RNA, nitrogenous bases between 2 nucleotides would form weak hydrogen bonds through a specific set of rules discovered by Irvin Chargaff in 1947. Looking back at the 4 possible bases for DNA and RNA, Chargaff's base pairing rules is as follows. Adenine pairs with thymine in DNA, while adenine pairs with uracil in RNA. And for both DNA and RNA, cytosine pairs with guanine. The understanding of the sugar-phosphate backbone and base pairs hints at the step of genetic expression and how both structures are formed.
Replication, transcription, and translation are the 3 steps that together form the central dogma of molecular biology. The first step, replication, refers to the process during which one DNA strand is duplicated. It is not only essential for biological inheritance, but it is also a necessity for cell division. The process of replication does not generate the same DNA strands. Rather, it creates a hybrid DNA with an old and new strand, hence it is a semi-conservative process. Before replicating, the double-stranded DNA is unzipped into 2 single-stranded DNAs. Helicase enzymes would break down the hydrogen bonds between nitrogenous bases, thus creating a Y-shaped formation called a replication fork. Topoisomerase enzymes are also applied to both strands to prevent supercoiling from disrupting the process. As discussed in the previous topic, the 2 strands of DNAs are anti-parallel, with one strand going in a 5 to 3 direction and the other one going in a 3 to 5 direction. Next, a primase enzyme is used to synthesize short RNA primers on each strand, which represent the starting position to build on. This allows the DNA polymerase enzyme to synthesize the new strand in the correct position. An important rule to note is that the DNA polymerase could only replicate in the 5 to 3 direction, which results in a leading and lagging strand. In the leading strand, a new strand is grown towards the direction of the replication fork. Therefore, only one primer is needed to carry out DNA polymerase's continuous route. However, in the lagging strand, the new strand is grown away from the direction of the replication, hence requiring multiple primers to constantly shift the DNA polymerase to a different position. This would result in a discontinuous strand, creating what's known as Okazaki fragments. Fortunately, the existence of ligase enzymes help ensure that the in-betweens are properly filled in to fix the new strand. After DNA replication is complete, the next step in the central dogma is the process of transcription. During transcription, DNA would be copied into mRNA. The mRNA would serve as a transcript for both genes and ribosomal proteins. During the initiation of transcription, a promoter pries open the 2 DNA strands, similar to helicase during DNA replication. A transcription start site is then indicated by the promoters, allowing the RNA polymerase to bind to it. Elongation is where most of the functional tasks of transcription occur. During this phase, RNA polymerase travels across the template DNA strand and employs base pairing methods with the DNA template in order to produce a new RNA copy. Similar to DNA replication, the reading of the template strands occur from the 5' to the 3'. Finally, the RNA polymerase would come in contact with the terminator, which makes the RNA polymerase detach from the DNA and release the newly synthesized mRNA, ending transcription. To finalize the mRNA, non-coding segments called introns are spliced, while the coding segments called exons are kept to create the final mRNA. Before we move on to translation, we must first explain the flow of information from gene to protein. Each set of 3 nucleotide bases in an mRNA form a codon, which represent a specific amino acid out of 20 possible ones. The group of specific amino acids would ultimately make up a protein. Since all organisms use the same genetic code, we can use a codon chart to easily identify the type of amino acid encoded. The nucleotide triplet, methionine, AUG, represents the start codon, which begins the translation process of the protein, whereas the nucleotide triplet of UA, UAG, and UGA represents the end codons, which ends the translation process of the protein. Here's an example of mRNA and its nucleotide bases. By using the codon chart, we could translate each triplet into a codon. Translation is the last component of the central dogma, where ribosomes in either the cytoplasm or the endoplasmic reticulum synthesize proteins. To begin, our RNA gather themselves around the mRNA. tRNAs are then brought into the rRNA, carrying amino acids from the cytoplasm, containing an anticodon, which are nucleotide pairs that read from 3 to 5. The tRNA can attach themselves to each triple nucleotide in the mRNA and encodes an amino acid to a growing peptide chain. The process is repeated until a stop codon is detected, in which the polypeptide is released from the tRNA to become a protein, ending the translation.
So here's an actual AP question regarding the translation of an mRNA to amino acids in the sickle cell anemia, which we will cover later during topic 6.7, which is about mutations. The HBS allele, which causes sickle cell disease, results from the mutation in the DNA sequence shown in figure 2, that produces a valine in place of a glutamic acid residue in a hemoglobin protein. Which of the following mRNA sequences is derived from the HBS allele? Well, using the codon table in figure 2 and the template strand as shown in the assignment, we could transcribe the mRNA. During the transcribing, we also have to make sure that we would leave one triple nucleotide pair as GUG due to the presence of valine in the mutation. Therefore, throughout all these processes, we would have obtained the answer D.
Each somatic cell contains the same DNA. Gene is regulated so that cells differ in their structure and function in different areas. Not all genes encoded in the DNA are transcribed into mRNA or translated into protein. The expression of specific gene is a highly regulated process with many levels and stages of control. This complexity ensures expression of each protein in the proper cell at the proper time. First of all, what is gene expression? Gene expression is the process of turning on a gene to produce mRNA and protein. It determines how much of the protein is made, and when it is time to stop making that protein. A regulatory sequence is a segment of a nucleic acid molecule which is capable of increasing or decreasing the expression of specific genes within an organism. Since prokaryotic organisms are single-celled organisms that lack a cell nucleus, their DNA flows freely in the cell's cytoplasm. When a particular protein is needed, the gene that codes for it is transcribed in the mRNA, which is simultaneously translated into protein. When the protein is no longer needed, transcription stops. As a result, the primary method to control how much of each protein is expressed in a prokaryotic cell is the regulation of transcription. The DNA of prokaryotes is organized into a circular chromosome that resides in the cell's cytoplasm. Proteins that are needed for a specific function or that are involved in the same biochemical pathway are only encoded together in blocks called operons. The genes in an operon are transcribed into a single mRNA molecule. This allows the genes to be controlled as a unit. Either all are expressed or none is expressed. Each operon needs only one regulatory region, including a promoter, where RNA polymerase binds, and an operator, where other regulatory proteins bind. In prokaryotic cells, there are three types of regulatory molecules that can affect the expression of operons. Activators are proteins that increase the transcription of a gene. Repressors are proteins that suppress transcription of a gene. Finally, inducer are molecules that bind to the repressor and inactivate them. Eukaryotic cells have intracellular organelles that adds to their complexity. In eukaryotic cells, the DNA is contained inside the cell's nucleus, where it is transcribed into mRNA. The newly synthesized mRNA is then modified and transported out of the nucleus into the cytoplasm, where ribosomes translate the mRNA into protein. The processes of transcription and translation are physically separated by the nuclear membrane. Transcription occurs only within the nucleus, and translation occurs only in the cytoplasm. The regulation of gene expression in eukaryotes can occur at all stages of the process. In eukaryotes, control of gene expression is more complex and can happen at many different levels. Eukaryotic genes are not organized into operon, so each gene must be regulated independently. In addition, eukaryotic cells have many more genes than prokaryotic cells. Regulation of gene expression can happen at any of the stages as DNA is transcribed into mRNA and mRNA is translated into protein. Regulation is divided into 5 levels: epigenetic, transcriptional, post-transcriptional, translational, and post-translational. Epigenetic changes are environment and other factor changes that can change the way genes are expressed. Epigenetic control involves changes to genes that do not alter the nucleotide sequence of the DNA and are not permanent. Instead, these changes alter the chromosomal structure so that genes can be turned on or off. This level of control occurs through heritable chemical modification of the DNA or chromosomal proteins. The best studied example of epigenetic regulation is modification of histone proteins. Histones are chromosomal proteins that tightly wind DNA so that it fits into the nucleus of a cell. The human genome, for example, consists of only 3 billion nucleotide pairs. An average chromosome contains 130 million nucleotide pairs, and each body cell contains 46 chromosomes. If stretched out linearly, an average human chromosome would be over 4 centimeters long. In order to fit all of this DNA into a nucleus of a microscopic cell, the DNA must be tightly wound proteins. This is also organized so that specific segments can be accessed as needed by a specific type of cell. The phenotype of a cell or organism is determined by the combination of genes that are expressed and the levels at which they are expressed. Observable cell differentiation results from the expression of genes from tissue-specific proteins. Induction of transcription factors during development results in sequential gene expression.
Once again, we have encountered yet another actual AP question, which focuses on the expression of genes. The figure above depicts a DNA protein complex that is assembled at the transcriptional start site of gene X when the expression of gene X is activated in liver cells. Previous studies have shown that gene X is never expressed in the nerve cells. Based on these diagrams, which of the following most likely contributes to the specific expression pattern of gene X? Based on what we previously learned on the regulation of proteins, some genes like operons from the prokaryotes cannot be expressed without the presence of activators. As not only do activators activate the transcription factors, but they also increase the rate of it. Therefore, it totally contributes to the expression patterns and how some genes were unable to be affected. Therefore, the answer is C.
Promoters are DNA sequence upstreams of the transcription start sites where RNA polymerase and transcription factors bind to initiate transcription. Negative regulatory molecules inhibit gene expression by binding to DNA and blocking transcription. Transcriptional regulation is control of whether or not an mRNA is transcribed from a gene in a particular cell, like prokaryotic cells. The transcription of gene in eukaryotes require an RNA polymerase to bind to a promoter to initiate transcription. In eukaryotes, RNA polymerase requires other proteins or transcription factors to facilitate transcription initiation. Transcription factors are proteins that bind to promoter sequence and other regulatory sequence to control the transcription of the target gene. RNA polymerase by itself cannot initiate transcription in eukaryotic cells. Transcription factors must bind to the promoter region first and recruit RNA polymerase to the site for transcription to begin. MicroRNA miRNAs has big role in gene regulation. MicroRNAs are a family of about 21 to 25 nucleotide small RNAs that negatively regulate gene expression at the post-transcriptional level. It binds to the RNA molecule and controls the stability of the mRNA in the cytoplasm. The longer an mRNA exists in the cytoplasm, the more time it has to be translated, and the more protein is made.
As discussed from the previous unit, genotypes determine the genetic makeup of all organisms, which are visually expressed through phenotypes. Additionally, the genotype is dependent on the genetic information provided by the DNA or RNAs to produce those genes. However, during gene expression, a slight alteration of DNA sequences could result from minimal to drastic changes to the amount of protein produced, and ultimately the phenotype expressed. These mutations are categorized into two broad groups: gene mutation and chromosomal mutation. Gene mutation happens when a single nucleotide is affected during a DNA replication. One type of gene mutation is substitution, where this one nucleotide is replaced by another. This would usually change the structure of the gene. An example of a substitution mutation is sickle cell anemia, where a glutamic acid, or GLU, is changed to valine, or VL, due to the substitution from andine to thymine, which is from GAG to GTG, which ultimately affects the hemoglobin gene. Sometimes during substitutions, the rRNA would still produce the same amino acid as before. Known as a silent mutation, it would still express the same phenotype, despite the different codons. The other type of gene mutation is a frameshift mutation, which is the insertion or the deletion of one nucleotide. Unlike substitution, a frameshift mutation would affect all amino acids, since the change of number of nucleotides would not only make them indivisible by 3, but it will also shift the reading frame. People with Tay-Sachs disease lack enzymes used to break down fatty substances due to the deletion of 1,510th nucleotide, which contained cytosine. That rendered the hexagene unable to provide instructions to produce the necessary enzymes. Chromosomal mutations focuses on the change of chromosomes or multiple chromosomes during mitosis or meiosis. The four main types are deletion, duplication, inversion, and translocation. In deletion, a segment of a gene of chromosome is lost. In duplication, a segment is repeated. In inversion, one segment is reversed, or tilted 180 degrees before attaching it back to the original chromosome. And in translocation, segments are shifted between two non-homologous chromosomes, which change the location of parts and of the genetic material. The final mutation occurs from physical, chemical, or biological regions known as mutagens. Harmful amounts of UV and X-ray radiation damages DNA by breaking the chemical bonds from sugar phosphate backbone and bases, leading to an unstable DNA structure. Soldiers exposed to nitrogen mustard gas during World Wars I and II not only received injuries to eyes and skin, but also produced new chemical bonds that prevented the double helix from unwinding ever again. This makes the DNA impossible to undergo replication, or transcription. Viruses are able to insert their own DNA to produce their own viral proteins. Whether they negatively or positively impact their survivability or reproduction of species, mutations are generally responsible for the genetic diversity of organisms. So here's a textbook question regarding gene mutations. The question asks which of the following mutations will be most likely to have a harmful effect on an organism. Based on what we learned on frameshift mutations, the deletion or insertion of nucleotides that are closest to the start of coding sequences would have had the greatest impact on the entire sequences. Since nucleotides after the change would be shifted and alternately paired to drastically different amino acids due to the different triple nucleotide pairs found. Therefore, the answer is D. From the studying of mutations and how they can affect the genetic structures and expressions of phenotypes within organisms, scientists began to search for techniques to manipulate the DNA in living cells, known as genetic engineering or biotechnology. The discovery of recombinant DNA technique in 1973 proved as the first major step into the introduction of biotechnology, since the discovery of the DNA structure in 1953. Cohen and Boyer, creators of the technique, has successfully replicated and expressed the phenotypes from the DNA of another organism. They first isolated a foreign DNA, then inserted into a plasmid of a bacterium. This turned it into recombinant DNA, which contains the DNA of two or more organisms. Finally, the plasmid is reinserted into the bacterium and was proved to be usable by the organism through further replication. A similar technique was further developed during the 1980s by Carl B. Mullis, which was able to produce billions of DNA copies within hours, known as the polymerase chain reaction. This technique requires a DNA sample, a Taq polymerase, which is a substitution to the original DNA polymerase, as well as nucleotides and primers. It contains three major steps: denaturation, which is the separation of two strands in DNA, annealing, which is the recombination of primers to each strand, and finally, elongation, which is the extension of nucleotides to the primers using the Taq polymerase. For each step, the temperature is adjusted from high to low to maximize the efficiency, enabling the process to repeat up to 35 times. With this new technique, clinics can now amplify certain genes to diagnose different genetic diseases, along with the further application into more new and interesting techniques such as gel electrophoresis. Gel electrophoresis is a technique used in genetic engineering to sort or analyze DNA segments based on their size and charge. Aragos gel, which is the gel used in this, which is extracted from seaweed, is placed in a box, along with wells which are filled with DNA samples. At the end of the box is a positive charge, which is used to pull the negatively charged DNA segments across the gel. The small-sized DNA fragments that are more negative would travel farther than the large-sized ones that are less negative, allowing you to separate between DNA fragments and molecules to measure the distance between each fragment. Often used for analytical purposes, it has also allowed scientists to not only characterize proteins and nucleic acids, but also catch criminals through DNA fingerprinting. With a variety of techniques to analyze and manipulate DNA and RNA, scientists are now able to genetically modify the lives of organisms and mass-produce medical products, all to maximize the continuity of life.